Welcome to another IB Environmental Systems and Societies video. Today's topic is 2.1 species and populations. In this video, we're going to examine how species interact with their environment, what defines their ecological niches, and how populations respond to various environmental factors. Understanding these concepts is really important for ecosystem analysis, and it forms the foundation of ecological studies. Let's get into it. Let's begin by defining some key terminology that's central to this topic. A species is a group of organisms that share common characteristics and can interbreed to produce fertile offspring. A population consists of organisms of the same species living in the same place at the same time and interacting with one another. The habitat refers to the environment where species normally lives, their physical home, so to speak. And a niche, or an ecological niche, describes the specific role that an organism plays within its habitat, including where it is, when it lives, how it lives, what it feeds on, and the other organisms that it interacts with. When we define a species, we need three key criteria: common characteristics, the ability to interbreed, and the production of fertile offspring. Let's look at dogs as an example. Despite their physical differences, all dog breeds share common characteristics. They can interbreed with each other, and they can produce fertile puppies. This means they're all members of the same species, Canis familiaris. Now, let's look at different species. Donkeys and horses share common characteristics and can interbreed, but their offspring, called mules, are infertile. That means that the mules cannot have offspring. Therefore, horses and donkeys are different species. Similarly, lions and tigers can produce hybrid offspring like ligers, but these hybrids are typically sterile as well. The inability to produce fertile offspring is a key indicator that we're dealing with different species. The habitat is simply the environment where a species normally lives. Think of it as an organism's address or physical location. This woodpecker's habitat is the forest environment shown in the image. The bird has specific adaptations that allow it to thrive in this habitat, such as its strong beak that it uses to drill into tree bark looking for insects. Habitats can be broadly categorized as terrestrial or land-based, or aquatic, and aquatic includes both freshwater and marine ecosystems. Each habitat type can be characterized by differences in the space, food, water, and shelter available. Examples of terrestrial habitats include deserts, mountains, and grasslands, while aquatic habitats include oceans, ponds, and estuaries. Each habitat has unique environmental conditions that support specific species adapted to those conditions. An ecological niche describes the particular set of abiotic and biotic conditions and resources to which an organism or population responds. The abiotic factors of ecological niches include physical elements like temperature, water salinity, and soil characteristics. Biotic factors include interactions with other organisms, such as food sources, predators, competitors, and beneficial organisms. This diagram illustrates how 5 different warbler species share the same habitat, coniferous trees, but they occupy different niches. Although they might appear to compete for the same resources, each warbler species feeds at a different height and a different portion of the tree. By partitioning the resources this way, they reduce direct competition with one another. This is a perfect example of how closely related species can coexist by occupying slightly different ecological niches. In the African savanna, we see another example of niche partitioning. Zebras, wildebeest, and gazelles all inhabit the same physical space but have different ecological niches based on their feeding behaviors. Zebras eat the tall grass first, then wildebeest consume what's left, and finally the gazelles come through to follow up and eat the new sprouts after the other animals have already moved on. This staggered feeding strategy reduces direct competition, and it allows all of these species to coexist in the same space. We need to distinguish between two important niche concepts. The fundamental niche describes the full range of conditions and resources in which a species could potentially survive and reproduce. The realized niche describes the actual conditions and resources in which a species does exist due to biotic interactions, particularly competition. This classic example shows how competition affects realized niches in barnacles. The diagram illustrates 2 different species that live on a rocky shore. Both species could theoretically live anywhere between high tide and low tide. That's their fundamental niche. However, abiotic conditions and competition with one another limit where each species actually lives. That's their realized niche. Looking more closely at the barnacles, Cathalamus can physically survive in both deep and shallow intertidal zones, which represents its fundamental niche. However, the Balanus outcompetes Thalamus in the deeper zone, restricting Thalamus to the drier upper portion of the intertidal zone. This restricted area represents Cathalamus's realized niche, the subset of its fundamental niche where it actually lives due to competitive interaction with the other barnacle species. Abiotic factors are the non-living physical elements that influence organisms and ecosystems. These include temperature, sunlight, pH, salinity, and precipitation. All these factors are interdependent. For example, sunlight influences temperature and evaporation rates, and those affect precipitation levels. Water availability impacts soil pH, which determines which plants can grow in a particular spot. Biotic factors involve the interactions between organisms. Predation is one such interaction, where one organism hunts and eats another one. In this image, we see an owl with its prey, a mouse. Predation is a critical biotic factor that shapes population dynamics and can influence the evolution of both predator and prey species. Herbivory is another important biotic interaction, especially the hunting of plants. Herbivory puts selection pressure on plants to develop defensive mechanisms like toxins or thorns, while herbivores evolve strategies to overcome these defenses. This evolutionary arms race is an ongoing process that shapes ecosystems. This close-up shows the impact of herbivory. The caterpillars are systematically consuming the leaf tissue, which can significantly affect plant growth and reproduction. Just as predation affects animal populations, herbivory can strongly influence plant populations and community composition in ecosystems. Parasitism is a biotic interaction where one species depends on another for nutrition, and that harms the host organism in the process. In this image, we see aphids feeding on a plant. Unlike predators that quickly kill their prey, parasites typically extract resources from their host over a long period of time, and this causes gradual harm rather than immediate death. Mutualism represents a biotic interaction where each species benefits from the relationship. This image shows a moth pollinating a flower. The moth receives nectar as food, while the flower achieves pollination necessary for reproduction. Mutualistic relationships are fundamental to many ecosystem processes, and they often result from long periods of co-evolution. Populations change and respond to interactions with their environment. This map shows how geographic isolation of an ancestral amphibian population led to speciation. Different environmental factors in each region selected for different characteristics that favored survival in those specific habitats. Over time, these isolated populations accumulated enough differences to become distinct species that no longer interbreed. Interactions between species should be understood in terms of how they influence population dynamics and carrying capacity. The diagram shows various factors that affect animal populations throughout the year. Wolf, moose, and lynx-hare relationships demonstrate classic predator-prey interactions. This is something you should definitely remember for your exam. Notice the cyclical patterns and the time lags in these population graphs. As prey populations increase, predator populations follow because they have more food, but they follow without a delay. The increased number of predators then reduces the prey population because they're eating the prey, and the prey population subsequently causes predator numbers to decline due to food scarcity. This cycle continues with remarkable regularity in natural systems. A population consists of organisms of the same species living in the same time at the same place and capable of interbreeding. This map of Tanzania shows different elephant populations represented by colored patches. These populations are separated by barriers, mostly areas with big human settlements and minimal elephant habitat. These barriers prevent regular interaction between the different populations of elephants. So even though all these elephants are the same species, they form distinct populations. Similarly, this map shows different Asian elephant populations across the continent. These populations are largely isolated from one another by geographic barriers in human development, and this also prevents genetic exchange. This isolation can amplify genetic abnormalities due to limited gene flow. While all Asian elephants belong to the same species, Elephas maximus, they are divided into separate populations that rarely interbreed. And it should be pointed out that Asian elephants and African elephants are two different species. Every ecosystem has a carrying capacity for a given species, the maximum population size that can be sustainably supported by the available resources. This diagram illustrates how animal populations fluctuate seasonally, with various factors keeping the population below carrying capacity. When populations exceed carrying capacity, resources become scarce, leading to increased mortality through starvation, disease, and other factors. Population growth curves can take two shapes, an S curve or a J curve. S and J population curves describe generalized responses of populations to environmental conditions. The J curve, or exponential growth, occurs when resources are abundant and few limiting factors exist. It also generally corresponds with very high reproductive potential. The S curve, or logistic growth, shows how populations initially grow rapidly, but then their growth rate levels off as they approach the carrying capacity. Abiotic factors like temperature, water availability, and space can limit growth, while certain biotic factors may promote it. Limiting factors slow population growth as it approaches the carrying capacity. The diagram shows several factors that can limit populations, including starvation, disease, parasites, accidents, weather, hunting, and predation. For example, during summer and autumn, elk populations might be limited by increased disease transmission from higher population density or by weather events like drought that reduce food availability. When interpreting graphical representations of ecological relationships, we observe important patterns. This graph shows the population cycles of snowshoe hares and Canadian lynx over 80 years. Note how the lynx population in red closely follows the hare population in the green. But the lynx population has a time lag. This demonstrates how predator populations respond to prey availability, and this is a classic example of population dynamics in action. Looking more closely at the lynx hare data, we can see the cyclical nature of these population changes. When hare populations peak, lynx populations increase because they have an abundant food supply. As the lynx numbers rise, they eat more hares, and that causes the hare population to fall. With fewer hares available to eat, lynx populations subsequently fall, and that allows the hare numbers to recover because there aren't as many lynxes out there to eat them. This starts the cycle anew. Every examiner will know this example. When explaining population growth curves, you have to consider both absolute numbers and rates of change. Exponential growth shows continuously accelerating growth rates, and that creates a J-shaped curve. Logistic growth begins exponentially, but slows as the population approaches carrying capacity, and that forms an S-shaped curve. The carrying capacity represents the maximum sustainable population size given an available resource base and environmental conditions. From an international perspective, changes in one ecological community can impact others, sometimes called the butterfly effect. Small changes in one system can cascade through interconnected systems, and that causes significant effects somewhere else. This underscores why environmental issues often require international cooperation and why ecological studies have to consider systems at different scales. That's it for ESS Topic 2.1, Species and Populations. In our next video, we're going to build on these concepts as we examine communities and ecosystems. Until next time, happy learning. Welcome to another IB Environmental Systems and Societies video. Today's topic is 2.2, Communities and Ecosystems. This important topic in ESS helps us understand how living organisms interact with each other and with their environment. Let's get into it. In ESS, a community is a group of populations living and interacting with each other in a common habitat. Think about a watering hole in Africa where zebras, elephants, and various antelope species all gather. They form a community by sharing the space and influencing each other's lives through competition, through predation, and other interactions. An ecosystem takes this concept even further by including both the community and the physical environment with which those communities interact. This coral reef ecosystem includes not just the turtle, the fish, and the coral organisms, but also the water, the sunlight, the ocean currents, the density of salt in the water, and all of those things shape the existence of the organisms that live there. Ecosystems are remarkably complex. This diagram shows a temperate forest food web with multiple interconnected feeding relationships. Notice how organisms participate in different food webs simultaneously. Some feeding on living plant material in a grazing food web, and others are consuming detritus in a decomposition food web. A significant idea in this topic is that photosynthesis and respiration play really important roles in the flow of energy through communities. This diagram illustrates how energy flows through an ecosystem while matter cycles within it. Energy enters as sunlight, it passes through various organisms, and it ultimately exits as heat. Photosynthesis and respiration are essentially opposite reactions. In photosynthesis, plants convert carbon dioxide and water into glucose and oxygen using light energy. In respiration, organisms break down glucose with oxygen to release energy, producing carbon dioxide and water as byproducts. When analyzing these processes as systems, you need to identify inputs, outputs, and transformations. For photosynthesis, inputs include light energy, carbon dioxide, and water. The outputs are glucose and oxygen. The transformation occurs within chloroplasts, converting light energy to chemical energy that's stored in glucose. Respiration can be described similarly as a system. The inputs to respiration are glucose and oxygen. Its outputs are carbon dioxide, water, and the energy released from respiration. The transformation occurs primarily in cellular mitochondria. Understanding these processes as systems helps us analyze the flow of energy through ecosystems. During respiration, large amounts of energy are dissipated or lost as heat, and this increases entropy or disorganization in the ecosystem. However, this energy release enables organisms to maintain their highly organized structures with relatively low entropy. This follows the second law of thermodynamics, which states that entropy in a system tends to increase over time. Primary producers, mostly plants and algae, form the foundation of ecosystems by converting light energy into chemical energy through photosynthesis. This stored energy becomes available to other organisms in the ecosystem when they're eaten. Some exceptional organisms, like bacteria near deep-sea vents, can produce food through chemosynthesis without using sunlight. Feeding relationships in ecosystems involve producers, consumers, and decomposers. These relationships can be modeled using food chains, food webs, and ecological pyramids. Each approach offers different insights into ecosystem structures and the way those ecosystems work. The primary producers form the first level, followed by primary consumers, the herbivores. The herbivores are followed by the secondary consumers, or the omnivores, who eat both herbivores and producers. Then you have your secondary consumers and your tertiary consumers, which tend to be carnivores that eat herbivores. At the side of each of these trophic levels, you have decomposers, and decomposers break down organic matter from all the trophic levels and recycling the nutrients back into the system and making them available for the producers. As energy moves through trophic levels, significant amounts are lost as heat. Following the 10% rule, only about 10% of the energy that's available at one trophic level gets passed on or transferred to the next trophic level. The remaining 90% is lost through metabolic processes, movement, and heat production through respiration. This inefficiency limits most food chains to typically 4 or 5 trophic levels. Bioaccumulation occurs when persistent pollutants like DDT or mercury build up within an individual organism over time because those substances can't be broken down or excreted by metabolic processes. This happens when the rate of intake of that substance exceeds the rate that it is eliminated or degraded. Biomagnification is related to bioaccumulation, but it's distinctly different. Biomagnification refers to the increase in the concentration of these pollutants as those pollutants move higher and higher up the food chain. Since each consumer eats many prey organisms, they accumulate all the pollutants those prey had stored, and that leads to increasingly dangerous concentrations at higher trophic levels. Toxins such as DDT and mercury accumulate along food chains due to this biomagnification process. This diagram shows PCB concentrations increasing dramatically from 0.000002 milligrams per liter in seawater to 160 milligrams per kilo in marine mammals. That's an 80 million-fold increase in concentration of PCBs. This high concentration of toxins and heavy metals in upper trophic levels is why a lot of doctors advise pregnant women and nursing mothers to avoid eating tuna and other top marine predators. Ecological pyramids are quantitative models that visually represent trophic structure. They typically show a decrease in numbers, a decrease in biomass, and a decrease in energy as we move higher and higher up the trophic chain. And this is what creates the pyramid shape. These are usually measured for a specific area and a specific time period. Let's go through these one by one. A pyramid of numbers simply counts organisms at each trophic level. Interestingly, these can sometimes be inverted, particularly when individuals at lower trophic levels are relatively large. For example, one tree, which is a producer, might support thousands of insects, which are the consumers, and those insects may support dozens of birds, which might then also support one or two predatory birds or other hunters like snakes. A pyramid of biomass represents the total mass of organisms at each trophic level, and this is measured in units like grams per square meter. These typically decrease at higher levels, but can occasionally show greater quantities at higher trophic levels if they're measured at a specific point in time, like during winter months when some of the producers in temperate or polar zones might be dormant. Pyramids of biomass can appear inverted during certain seasons, especially in aquatic ecosystems. This diagram shows how a biomass pyramid changes through seasons as producer populations fluctuate. However, if it's measured over an entire year, the pyramid would show the expected decreasing pattern, resulting in that pyramid shape. Pyramids of productivity show the flow of energy through each trophic level. That's the rate at which biomass is generated. You can identify these by the time component in their units, such as kilos per hectare per year, or grams per square meter per year. When you see that per year, you're probably looking at a pyramid of productivity. Unlike biomass pyramids, productivity pyramids for entire ecosystems over a year always show a decrease along the food chain. You can calculate the efficiency of energy transfer between trophic levels. In this example, of the 1,575 grams per square meter per year that's produced by plants, only 235 grams of biomass per square meter per year becomes herbivore biomass. That's an efficiency of about 15%, and is relatively close to that 10% rule we addressed earlier in the video. The efficiency generally declines at higher trophic levels due to increasing energy demands for maintenance. In other words, your top predators spend a greater proportion of their energy hunting for moving organisms than the herbivores do wandering the landscape and eating plants that don't run away from them. The laws of thermodynamics govern energy flow in ecosystems. The first law of thermodynamics, which is called the conservation of energy, explains why energy is neither created nor destroyed, but it's transformed or it changes forms as it moves through food chains. The second law of thermodynamics, which is the law of increasing entropy, explains why energy becomes less available with each transfer, because much of that energy is lost as heat. A key skill in ESS is constructing models of feeding relationships from data. You might be asked to do this in one of your paper 2 questions in section A. This table shows population changes in a marine ecosystem over time. You could use this to build food chains or food webs or ecological pyramids showing relationships between kelp, urchins, sea otters, and other species. When constructing system diagrams for processes like photosynthesis and respiration, make sure you include components such as flows, inputs, outputs, transfers, transformations, and storages. And remember on the exam to always write those system diagrams as boxes and arrows. They're not looking for a sketch of the organisms. You also want to remember that energy flows through a system while matter cycles within it. Many ecosystems cross political boundaries, and this creates international management challenges. Questions arise about who controls water resources, who's responsible for enforcement, who pays for cleanup, and who compensates those who are affected downstream from pollution. From a TOK perspective, it's worth considering how different models represent feeding relationships. When is a food web more appropriate than a simple food chain? When might a pyramid of numbers be more informative than a pyramid of biomass? These kinds of choices influence the way that we understand ecosystem dynamics. That's it for ESS Topic 2.2, Communities and Ecosystems. Thanks for watching, and until next time, Happy learning. Welcome to another IB Environmental Systems and Societies video. Today's topic is 2.3 flows of energy and matter. In this video, we're going to examine how ecosystems are linked through energy and matter flows and how human activities impact these vital processes. Let's get into it. Ecosystems are fundamentally connected by energy and matter flows. Remember the food webs we studied in topic 2.2? Those illustrate how energy flows from producers to consumers and to decomposers. Chemical cycling, shown by the blue arrows, represents how matter moves through the ecosystem over and over again, while energy flow, shown by the red arrows, is one directional. It enters the ecosystem, transfers, transforms, and then ultimately exits the system. The sun's energy drives these flows, and humans are impacting them both locally and globally. This diagram shows how solar energy is distributed through Earth's systems. Only a tiny fraction, about 0.08%, is captured by photosynthesis, while most of it becomes thermal energy. Notice the human use component, which taps into fossil fuels. This is ancient solar energy that's been stored for hundreds of millions of years. As solar radiation or insolation enters Earth's atmosphere, much of it becomes unavailable for ecosystems. This energy budget shows how incoming solar energy is reflected by the atmosphere, clouds, and Earth's surface, while the remainder is absorbed by land, oceans, and the atmosphere. Looking more closely at radiation pathways, we see that about 31% of incoming solar radiation is reflected and not absorbed at all, through processes like cloud reflection, ground reflection, and atmospheric scattering. The other 69% is absorbed by Earth through land, water, atmospheric, molecules, and clouds. Energy follows specific pathways through ecosystems. First, light energy is transformed into chemical energy through photosynthesis. This chemical energy transfers between trophic levels with varying efficiencies. And eventually, all that energy is transformed to heat and re-radiated to the atmosphere and ultimately out into space. Here, we see energy transfer between trophic levels. Notice how only about 10% of the energy moves from one level to the next. The rest is lost as heat. This energy pyramid shows primary producers capturing 100% of available energy, with subsequent levels retaining progressively less. A significant portion of solar energy is converted to heat. In this Earth energy budget, we can see how absorbed light is eventually re-radiated as infrared energy. This transformation follows the second law of thermodynamics, with energy becoming less concentrated and available. The final pathway for energy is re-radiation of the heat to the atmosphere. Note the large red arrows showing energy leaving Earth's surface. This release of heat energy is really important for maintaining Earth's temperature balance, and it's what drives weather patterns and ocean currents. Productivity measures the conversion of energy into biomass over time. It's always a rate, and it's typically measured in grams per square meter per year, but sometimes you'll see it in kilos per hectare per year for terrestrial ecosystems. This diagram shows how biomass decreases at higher trophic levels due to energy losses between transfers. Net primary productivity is abbreviated as NPP, and gross primary productivity is abbreviated as GPP. GPP represents the total biomass produced through photosynthesis, while respiration is the energy that is used by producers to maintain their living function. NPP is what remains. It's the actual growth that becomes available to the other organisms that eat those producers. Similarly, gross secondary productivity, abbreviated as GSP, is calculated by subtracting fecal loss, that's loss from pooping, from food eaten by consumers. It represents the total energy assimilated by consumers. Mr. G's ESS website has some excellent resources explaining these concepts if you want to check that out. Net secondary productivity, abbreviated NSP, equals GSP, or gross secondary productivity, minus respiratory losses. This represents new biomass created at consumer levels, the actual growth of consumer populations after accounting for the energy that they used in respiration. It's just like the productivity at primary levels, except we're talking about consumers instead of producers. Maximum sustainable yields are equivalent to the net productivity of a system. Matter also flows through ecosystems, involving transfers or movements from place to place, and transformations, which is a change in state or a change in form. A sustainable yield is the amount of biomass that can be removed from the system without diminishing the system's natural regeneration rate. For example, let's consider these lions in Tanzania. If we've got 1,000 lions and there are 78 lions born every year, and then approximately 48 lions die natural deaths every year, the natural income is 30 lions per year. Theoretically, 30 lions could be sustainably harvested or hunted every year without reducing the population, although the optimal sustainable yield is typically about 50% of this to account for uncertainties such as a new disease outbreak or natural hazards or storm events that kill members of the population unexpectedly. The carbon and nitrogen cycles illustrate matter flows using diagrams that show storages or sinks and flows. In the carbon cycle, carbon transfers between locations and transforms between states. It goes from gaseous CO2 to solid biomass or fossil fuels. Storages in the carbon cycle include both organic forms, as in organisms and forests, and inorganic forms, such as the carbon that you find in the atmosphere or in soils or fossil fuels and in the oceans. Each storage holds carbon for different amounts of time, from days in organisms to millions of years in fossil fuels. Flows in the carbon cycle include consumption or feeding, death, and decomposition, photosynthesis, respiration, dissolving, and fossilization. These processes move carbon between different reservoirs or sinks in the biosphere. Similarly, the nitrogen cycle includes organic storages in organisms and inorganic storages in soil, fossil fuels, the atmosphere, and bodies of water. Most nitrogen exists as atmospheric nitrogen gas, which most organisms can't use directly. Flows in the nitrogen cycle include nitrogen fixation by bacteria and lightning, absorption, assimilation, consumption, excretion, death, decomposition, and denitrification. These processes convert nitrogen between forms that organisms can use and forms that they can't use, and back to forms that they can use. Human activities significantly impact energy flows and biogeochemical cycles. This energy flow diagram shows sources and uses of energy in the United States, with fossil fuels dominating. Consider how these energy choices affect carbon and nitrogen cycles through inputs, outputs, and storage changes. When you're analyzing quantitative models of energy flow, you want to look for patterns in that numerical data. What energy sources predominate? In other words, what are the main ones? How efficiently is energy transferred between sectors? How might these patterns impact ecosystems through carbon emissions or land use changes? When you're constructing quantitative models of energy or matter flows, you want to make sure that you include all the major components with their numerical values. This example shows energy flow through a lake ecosystem with values in kilocalories per square centimeter per year. Analyzing the efficiency of energy transfer typically requires calculating percentages. These ecological pyramids show how biomass and energy decrease at higher trophic levels. In the energy pyramid at the bottom, only 16% of producer energy reaches primary consumers at the next level. To calculate gross primary productivity and net primary productivity, or GPP and NPP values, you want to use the formula NPP equals GPP minus R. Gross secondary productivity and net secondary productivity can also be calculated using a similar but slightly different formula. GSP equals the food eaten minus the fecal loss, and NSP equals the gross secondary productivity, or GSP, minus R for respiration. Understanding these calculations help quantify energy flow through ecosystems. The formulas themselves are pretty easy to use. The trick is recognizing which data goes with which abbreviation. The trick is recognizing which data point goes with which abbreviation. If you're asked to discuss human impacts on energy and matter flows on your exam, you want to consider how agricultural practices, fossil fuel use,
deforestation disrupt natural cycles. Harvesting biomass removes energy that's stored at molecular binds. Fossil fuel extraction occurs much faster than those fossil fuels are formed. Nitrogen-rich crop removal depletes soil nutrients unless they're replaced through natural soil amendments or commercial synthetic fertilizers. Finally, consider the international perspectives and philosophical questions. Human impacts on energy and matter flows cross borders. Emissions in one country affect climate globally. Deforestation can impact atmospheric circulation patterns, and overfishing depletes ocean food webs worldwide. Throughout history, cultures have recognized the sun's importance through mythology. These stories sometimes preserve knowledge before scientific understanding was written down. That's it for ESS Topic 2.3. Next time, we'll explore biomes, zonation, and succession in Topic 2.4.
Welcome to another IB Environmental Systems and Societies video. Today's topic is 2.4, Biomes, Zonation, and Succession. We'll examine how climate determines biome distribution, how communities change across environmental gradients, and how ecosystems develop over time. Let's get into it. The key concept for this topic is that climate largely determines what type of biome we find in any given area. This is illustrated in the graph showing average annual temperature and precipitation. Notice how different biomes occupy different regions of this climate space. While climate sets the stage, individual ecosystems within biomes can vary considerably due to local abiotic factors like soil type and biotic factors like the specific species that are found there. Biomes are collections of ecosystems that share similar climatic conditions and can be grouped into 5 major classes: aquatic, forest, grassland, desert, and tundra. Aquatic biomes are water-based, and they include both freshwater and marine ecosystems. Each biome class has characteristic limiting factors that constrain or limit growth, and they have different levels of productivity, and they have different levels of biodiversity. Terrestrial biomes are land-based. If you look at the world map, you can see how these biomes are distributed globally. Notice the patterns. Similar biomes often occur at similar latitudes around the world. That's their distance from the equator. This is because climate follows predictable patterns based on latitude, which is we're going to explore in just a few moments. Here we see a specific example of an aquatic biome. This slide highlights how aquatic biomes, like all major biome classes, have specific limiting factors, productivity levels, and biodiversity characteristics that make them distinct. For aquatic ecosystems, limiting factors often include light penetration through water and the availability of dissolved nutrients. Forests are biomes dominated by trees. Forest biomes have high primary productivity, but they can be limited by nutrient availability, especially in tropical regions where nutrients are rapidly cycled and primarily stored in the biomass of the trees rather than in the soil. Grasslands are characterized by grass as the dominant vegetation type, with just a few trees. These ecosystems receive less precipitation than forests, their productivity is moderate, and it can be limited by seasonal temperature extremes and relatively slower nutrient cycling. Desert biomes receive very little precipitation, and they experience extreme temperature fluctuations between day and night. The limiting factor in deserts is primarily water availability, and that restricts photosynthesis. Despite these harsh conditions, desert soils can be rich in nutrients due to minimal leaching from precipitation. Tundra biomes are characterized by extremely cold temperatures, short growing seasons, and often permafrost. That's permanently frozen ground. Limiting factors include short days for part of the year, low temperatures, and frozen water that restricts plant access to moisture even when it's present in the ecosystem. All of these things limit photosynthesis in the tundra. This table summarizes the specific limiting factors for each biome type. For aquatic biomes, light absorption by water limits photosynthesis, especially in deep oceans. Forests face constraints like nutrients locked in biomass instead of in soil. Grasslands have less precipitation and temperature extremes. Deserts lack water, and tundras face short days and frozen conditions that limit productivity. Looking at productivity across biomes, we see significant differences. Tropical coral reefs and rainforests show very high productivity, while deep oceans and deserts have low productivity. Note how productivity relates to the availability of resources and energy for photosynthesis in each biome. Biodiversity also varies greatly across biomes. Tropical rainforests have the highest biodiversity on Earth, with temperate forests coming in a close second. Desert and tundra biomes typically have lower biodiversity due to their harsh environmental conditions that fewer species have adapted to survive. Let's examine what creates these biome distributions. Insolation, which is incoming sunlight, precipitation, and temperature are the main factors governing where biomes occur. Insolation refers to the amount of solar radiation reaching Earth's surface, and that varies by latitude or distance from the equator because of Earth's tilt. This difference in insolation, combined with differences in how land and water absorb heat, creates the temperature patterns we observe globally. This triangular diagram shows how the interaction of temperature, precipitation, and evapotranspiration determines which biome will develop in which location. Moving up the diagram represents increased moisture, while moving right represents increases in temperature. Each combination of these factors creates conditions that are suited for a specific biome type. The tricellular model of atmospheric circulation is something you should definitely know for your ESS exam. The tricellular model explains the distribution of precipitation and temperature globally. There are 3 cells in each hemisphere that you need to know about. The Hadley cells near the equator circulate warm air. The Ferrel cells in the mid-latitudes circulate moderate temperature air. And third, the polar cells circulate cold air. This circulation pattern creates predictable climate zones, and those climate zones influence the distribution of biomes. Climate change is altering the distribution of biomes. This image shows projected changes in tree biomass, grass biomass, and total biomass across Africa between the years 2008 and 2100. Notice the significant shifts predicted, particularly in the map on the right. These changes occur as warming temperatures alter precipitation patterns, and they push the climate zones towards the North and South poles, or away from the equator. Zonation refers to changes in community structure along an environmental gradient. Unlike succession, which is a change over time, zonation is a spatial phenomenon where you can observe different communities as you move across the landscape from point A to point B. This coastal example shows how distinct zones develop from subtidal channels to the high marsh habitats. Here's an excellent example of altitudinal zonation. As we move up a mountain, both moisture and temperature change, and that creates distinct vegetation zones along the mountain. Notice how ecosystems at higher elevations resemble those found at higher latitudes. Moving up a mountain is climatically similar to moving towards the North or South Pole. It's important to understand the difference between zonation and succession. Zonation is a spatial pattern observed across the landscape at a single point in time. Succession is a temporal change where communities evolve and change over time. The image shows vertical zonation on a mountainside, illustrating how elevation changes create distinct ecological communities. This significant idea introduces succession, which is the process that leads to climax communities. Succession proceeds through stages or sears, from pioneer species colonizing bare rock to increasingly complex communities. The diagram shows the progression from bare rock through various sears to a forest climax community. There are 2 main types of succession. Primary succession starts from bare rock or substrate with no prior soil development, where no organisms have lived before. This usually happens after a volcanic eruption. Secondary succession occurs after a disturbance to an existing ecosystem, like a fire or a flood, where some soil and organic matter remain behind. Secondary succession generally proceeds a lot faster since some resources and nutrients are already present on site. Succession moves through distinct stages over time. Those stages are called sears, S-E-R-E. We start with a climax community at the top, which may be disrupted by disturbance. Pioneer species then colonize the area, followed by intermediate stages with increasing complexity, eventually returning to a climax community if left undisturbed for a long enough period of time. During succession, several ecosystem properties change predictably. This graph shows how productivity, biomass, stability, and diversity all change over time following a disturbance. Note that productivity peaks relatively early in succession, while stability continues to increase throughout the process. Greater habitat diversity leads to greater species and genetic diversity. This relationship occurs because more diverse habitats provide more environmental niches, and those can support more specialized species. The graph shows a strong positive correlation between habitat diversity and bird species diversity found in Nevada. Species can be classified based on their reproductive strategies and potential. Our strategists thrive in unstable environments with frequent disturbances. They produce a lot of offspring, they mature quickly, and they tend to have short lifespans. K strategists, on the other hand, are found in stable environments. K strategists produce fewer offspring, they grow more slowly, and K strategists tend to invest more resources in ensuring the survival of each offspring. This image illustrates the RK scale of reproductive strategy. On the extreme R selected end, organisms like oysters produce millions of offspring annually. On the K selected end, species like chimpanzees might produce just one offspring every 5 years. These strategies represent adaptations to different ecological niches. These graphs show different population dynamics of R and K selected species. R selected species show large population fluctuations. They often overshoot the carrying capacity and then experience a mass dieback. K selected species maintain more stable populations that hang out near the carrying capacity. The survivorship curves also differ dramatically, with R selected species experiencing high mortality or a lot of deaths early in life. In early stages of succession, gross productivity starts low due to the unfavorable conditions and the low density of producers. However, community respiration is also low, and that results in high net productivity as the system accumulates new biomass. The graph shows how gross production rises quickly, then gradually decreases while biomass continues to increase. In later succession stages, with an increased consumer community, gross productivity may remain high, but it's balanced by higher respiration rates. The productivity to respiration ratio, or P to R ratio, approaches 1, and that means that net productivity approaches 0. At this stage, the ecosystem is maintaining its biomass rather than accumulating more. This diagram illustrates how ecosystem stability, succession, and biodiversity are intrinsically linked. It shows the progression from bare rock through pioneer stages to climax forest, with increases in biodiversity, biomass, and soil development occurring throughout the entire process. In a complex ecosystem, the variety of nutrient and energy pathways contributes to its stability. This food web diagram shows 56 species with numerous connections between them. Each connecting line represents energy flow and can be thought of as a supporting structure. The more connections that exist, the more resilient the whole system comes to disturbances. There is no single climax community, but rather there's a set of alternative stable states for a given ecosystem. This forest shows patches of different colors representing different communities within what's considered a climax ecosystem. These variations arise from differences in soil conditions, in microclimate, and past disturbance events like storms that may hit one part of the ecosystem and leave other parts of the ecosystem alone. Human activity can divert succession to alternative stable states by modifying ecosystems. Activities like agriculture, grazing, fire management, or deforestation can create what ecologists call a plagioclimax community. A plagioclimax is a stable state that's maintained by continued human intervention. If human activity stops, as in this field in Denmark, secondary succession will begin. An ecosystem's capacity to survive change depends on its diversity and its resilience. Tropical rainforests, despite their high biodiversity, recover pretty slowly from disturbance because most of the nutrients are stored in vegetation rather than in the soils. Temperate forests, on the other hand, often recover more quickly because they have more nutrients in the soil to support regrowth when the plants are cut down. To explain biome distributions, you have to consider temperature, precipitation, insolation, and global air circulation patterns. This diagram shows how these factors interact to create the climate conditions that support different biomes. Note how both latitude and altitude influence biome type due to their effects on temperature and precipitation. Analyzing biome data reveals how climate factors influence flora and fauna adaptations. That's plant and animal adaptations. For example, tundra environments with really cold temperatures and minimal precipitation support specialized plant species like mosses and lichens, and animals that are adapted to cold like caribou and lemmings. Each biome's characteristic species show adaptations to the specific climatic constraints within that biome. Climate change is impacting biomes around the world. These maps show temperature changes from 1901 to 2002, biome distributions from 1961 to 1990, projected biomes for 2071 to 2100, and vulnerability to biome change. Areas with high vulnerability, those are in red, face pretty significant ecological transformations as climate zones shift towards the north and south poles and upward in elevation or altitude. Soil formation is the foundation of primary succession. This diagram shows the process from weathering of bare rock to simple organism colonization to horizon formation to well-developed soils that can support increasingly complex plant communities. Each stage or sear of succession creates conditions that facilitate the next stage. This is an example of a positive feedback mechanism in ecosystems that's actually good. Succession follows predictable patterns of community change. This progression shows increasing species richness and evenness as succession advances. Pioneer communities typically have low diversity and they're dominated by a few species, while climax communities have higher diversity with more evenly distributed species populations. Various factors can lead to alternative stable states within an ecosystem. This forest shows patches of different vegetation communities coexisting at the same time. These differences might result from variations in soil conditions, maybe some changes in microclimate or differences in microclimate from one side of a hill to another, past disturbance events like fires or storms, or historical contingencies, especially which species happened to arrive first. There are important links between ecosystem stability, succession, diversity, and human activity. Human activities that reduce biodiversity often decrease ecosystem stability. Practices that interrupt succession, like frequent clearing, prevent ecosystems from developing the complex food webs that contribute to resilience against disturbances. R and K selected species play different roles in succession. R selected species, with their fast growth and their high reproductive rates, dominate the early stages of succession. Their ability to quickly colonize disturbed areas makes them really effective pioneers. The K-selected species with slower growth and more competitive abilities tend to dominate later succession stages and the climax communities. This sand dune system provides an excellent opportunity to interpret a model showing both succession and zonation. This is primarily zonation because we see spatial changes moving inland from the shore. However, it also represents succession over time, as younger dunes near the shore eventually develop into the older, more complex communities found inland. From an international perspective, zonation occurs at both local and global scales. The same principles that create zonation along a small transect also operate at continental scales. From a TOK perspective, studying ecosystems requires decisions about which factors to measure. The challenge lies in determining which variables are most significant before fully understanding the system. That's it for topic 2.4, biomes, zonation, and succession. Remember that these concepts help us understand how ecosystems are distributed globally, how they change across landscapes and time, and how they develop throughout the years. Welcome to another IB Environmental Systems and Societies video. Today we're looking at ESS topic 2.5, investigating ecosystems. This section of the ESS course focuses on the practical methods you're going to use to study ecosystems. Understanding these techniques isn't just about passing your exams. These are the same tools that scientists use worldwide to monitor environmental change and develop conservation strategies. They're also really useful for your internal investigation. So we're going to explore how to quantify, analyze, and understand the natural world. Let's get into it. The first significant idea here is fundamental. Ecosystem investigations allow us to make comparisons between different ecosystems and track changes over time. Think about why this matters. Without standardized methods to study wetlands like the one shown, we can't determine if habitats are improving or degrading, and we can't understand how human impacts are changing natural systems around the world. Our second significant idea emphasizes quantification. Nature doesn't arrange itself neatly for our convenience, so we impose measurement systems on it. When you count plants or measure soil pH, you're converting the messy complexity of nature into data that can be analyzed. This transformation from qualitative observation, that's observation that you describe with words, to quantitative data, that's numerical data, is what allows us to draw meaningful conclusions about environmental patterns and processes and human impacts on them. Every ecosystem investigation has to start with proper identification. Just as you have a name and address, ecosystems need precise names and locations too. This allows other scientists to find your study site, to replicate or repeat your work, or track changes over time. The specificity matters. Saying a forest in Switzerland is really different from identifying Dinkerwald, a mixed deciduous coniferous managed woodland in Bar, Switzerland. The second one is a lot more precise and allows other people to find and repeat your work. Organism identification is essential for ecological studies. Dichotomous keys are brilliantly simple tools. Each step presents just two options, gradually narrowing possibilities until you identify your specimen. Those options are generally a yes or a no question. Remember that effective keys use clear comparative characteristics rather than subjective terms. A good key might ask, does the leaf have a serrated edge? Rather than, is the leaf pretty? This more complex dichotomous key for river macroinvertebrates shows how keys work in practice. Notice how the structure branches repeatedly, with each decision point narrowing the possibilities. This systematic approach transforms identification from guesswork to science, and that allows even non-specialists to accurately identify species. This is an important skill for ecosystem monitoring and assessment. Sampling strategies allow us to study large areas by examining representative parts of that area. This slide shows how we might measure changes in space or along gradients over time, through succession, or before and after human impacts. Quadrats are among our most fundamental tools for environmental monitoring. These simple frames define precise areas where we can count organisms or measure environmental factors. Transects are another essential sampling technique, and they allow us to study how conditions change across environmental gradients. The point transect shown here records data at specific points along a line. This method is perfect for studying zonation, how species distribution changes with distance from a shore, up and down an elevation on a mountain, or along other gradual environmental transitions. Belt transects combine benefits of quadrats and point transects. By placing quadrats at intervals along a transect line, you can get detailed information at regular distances. This technique excels at capturing how community composition shifts or changes across boundaries like forest edges, shorelines, or pollution gradients. This is precisely the kind of transition that often interests environmental scientists the most. How we arrange our samples dramatically affects our results. Random sampling prevents bias, but it might miss important patterns. Systematic sampling ensures even coverage, but could coincide with environmental patterns. Stratified sampling targets different habitat types deliberately. Each one of these approaches has strengths and limitations, and your research question should determine which method you choose. Here's a real quadrat in action, showing how this simple square frame creates a defined study area. Notice how it captures a small, manageable section of the ecosystem that can be thoroughly inventoried. The power of quadrats comes from their repeatability. By using the same method in the same way in different locations or times, you can make valid comparisons across space and time. Scientific conclusions are only as good as the data they're based on. Repeating measurements increases reliability by reducing the impact of random errors or anomalies. The number of repetitions needed varies by factor. Highly variable measurements like species counts might need many repetitions, while stable factors like soil type might need fewer repetitions. Measuring biomass helps us understand energy flow through ecosystems. The process requires careful sampling, drying to remove water content, and extrapolation to estimate total biomass. Notice the challenge mentioned. What about root biomass? This highlights how invisible components of ecosystems are frequently overlooked, but they may represent pretty significant portions of the total biomass and ecosystem function. Organisms can either be fixed in place, like vegetation, or they can move around. We call those organisms that move around motile organisms. In the US, a lot of times you'll see mobile organisms. For non-motile organisms like plants, we have several measurement techniques. Actual counts work for larger, distinct individuals, like trees. Percentage cover estimates the proportion of area covered by a species. That's useful for grasses and mosses. Percentage frequency records how often a species appears in sample plots. Each method suits different research questions and the type of organism. Estimating populations of moving organisms presents unique challenges. Direct methods like visual counting can be accurate, but that's often labor intensive and it's impractical for large areas or elusive species that are hard to find. The tagged dragonfly illustrates how scientists directly mark individuals for later identification. That's part of a more sophisticated population estimation technique. The Lincoln Index provides an elegant solution for estimating wildlife populations. By capturing, marking, and recapturing organisms, we can calculate total population size without counting every individual. This is the method that you should remember for estimating population size. Capture, mark, recapture is called the Lincoln Index. The formula looks complex, but it follows simple logic. The proportion of marked animals in the second sample should reflect the proportion of marked animals in the whole population. For your exams, you don't need to memorize the formula because it will be given to you, but you absolutely must know how to use and apply it. Species richness is simply the count of different species present. That's a fundamental biodiversity measure. The illustration contrasts two communities with identical species richness, but they have different species evenness. Community 1 has balanced populations, while Community 2 is dominated by a single species. This shows why richness alone doesn't tell the complete story of biodiversity. Species diversity indices can indeed feel overwhelming. The key point is that diversity combines both richness, the number of species, and evenness, the relative abundance of those species. Think of diversity indices as mathematical tools that compress complex community data into single, comparable numbers. That's important for monitoring changes over time. This visual perfectly illustrates the difference between species richness and species evenness. Both communities contain 4 tree species, but Community 1 has equal numbers of each, that's high evenness, while Community 2 is dominated by yellow trees. That means that Community 2 has low evenness. This distinction matters ecologically. Diverse, even communities usually show greater stability and resilience to disturbance. The Simpson Diversity Index formula calculates diversity based on both richness and evenness. Higher index values indicate greater diversity. Remember that this index is most valuable for comparing similar habitats or tracking changes in one location over time. The formula may seem intimidating, and you don't need to memorize it for the exam because, like the Lincoln Index, you'll be given the formula. But calculators and spreadsheets can make application pretty straightforward, and the numbers you'd be given on your ESS exam will be simple enough that you can do it in just a few minutes. The most important thing is that you know how to use the Simpson Index and when to use it. Ecological investigations form the perfect basis for your ESS internal assessment, or IA. Notice how the components align perfectly with IA requirements, identifying the context, planning your investigation, analyzing your results, evaluating the methods that you used, applying your findings, and of course, clear communication using scientific terminology. A well-designed ecosystem investigation shows your environmental understanding while developing transferable research skills that you can use elsewhere. Creating identification keys is a practical skill you should practice. It will help you understand how to use a dichotomous key if you see one on your exam. When making keys, focus on obvious, permanent features that clearly differentiate species. Avoid subjective descriptions like beautiful or large in favor of measurable characteristics like leaves over 10 centimeters long or stems with visible thorns. Critical evaluation of sampling strategies demonstrates higher level thinking. Consider how random sampling prevents bias, but it might also miss rare species, while systematic sampling ensures coverage of the entire area, but it might coincide with environmental patterns. The best strategy depends on your research question, the available resources, and the ecosystem being studied. These TOK connections highlight important questions about the method, validity, and scope of investigations. When is quantitative or numerical data superior to qualitative descriptive observations? How does knowledge gained through field observation compared to controlled lab experiments? These kind of questions invite us to consider how we know what we claim to know about environmental systems. That's it for ESS Topic 2.5, Investigating Ecosystems. You should be able to describe these ecosystem investigation techniques and when to apply them for success on your exams. These are the same methods used by scientists worldwide to monitor environmental change, to develop conservation strategies, and to understand our changing planet. Good luck, and until next time, happy learning.